Good afternoon, grade 12s, and welcome to today's lesson on quarter one work. We will be doing some revision on everything that you've completed already. My name is Yolanda Gibson. I'm a teacher at Parklands College, and I will be helping you guys through some of the resource material, the tutoring material that you would have received from your teachers today. So let's dig into it. Right, so during the exams, you guys will be receiving the usual format that you're accustomed to at your own schools, where one of the question types might be based on a multiple choice question. So that's your question 1.1 section. And it's important when we get to this type of question that we read what the question is asking. So there are various different ways to approach a multiple choice question, and I will be helping you guys through two of the ways in which usually ends up getting you close to the answer. So let's have a look at it. Now, the first step would be to always make sure that you read the story, the stem of the question, and then cover your four possible answers. Think about which one could possibly be the correct answer, and then uncover the four possible answers. So here we've got our four possible answers, and if you immediately see the correct answer, you go and circle it. But let's say, for instance, that you're unsure which of the four options are correct. Then make use of process by elimination, so you go for those that are definitely incorrect, and the one that seems to be the most correct answer, that will be your option. But remember to never leave a multiple choice question unanswered. So you will end up having a 1 in 4 chance of getting it right. So you're playing a little bit of a lotto there, so make sure that you always answer all the questions throughout the paper. So when you're done with your paper, go back, read, go through your answers, make sure that everything has been filled in. Right. So here we're going to look at a typical paper question that you will experience in grade 12, and they ask us which one of the following is true about genes. So genes are, now in order to answer this question, they want to know what is a gene, and they will ask which one is true. So out of the four possible answers that will be given, we want to know which one is correct from the statements. But let's look at the definition for a gene first. So gene is a segment of DNA, or it can be a segment of a chromosome that is responsible for a particular characteristic. And obviously in here we've got an example, it could be like attached or unattached earlobes. So when I look at the image that I have here on the side, that portion would be an example of a gene. So all those nitrogenous bases that we see here, that could potentially code for one characteristic, and then the nitrogenous bases just underneath it, that could be for another characteristic. So our example could be the top one could be for hair color, for instance, and the bottom one could be for an earlobe, for instance. Right, so now that we've got the definition in front of us, let's look at our four possible answers that will be given to us. If we read the first one, it says codes for a particular characteristic. And if you have memorized your work, your terminology, which is obviously very important because there could be up to 10 marks just allocated for your terminology, then this would have been quite an easy answer for you to get to. So remember not to skip on your terms when it comes to studying. Right, so from the options that we have, the correct answer then would be A, codes for a particular characteristic. But if we look at the rest of the options, why they are incorrect, if we go to B, we see the answer there is made up of amino acids, but we know that amino acids are the building blocks to proteins. C, made up of RNA, but genes make up DNA. RNA are your examples of your tRNA or your mRNA. And then for D, it says found in the ribosome, but we know that DNA is found inside your nucleus. So that one is obviously then incorrect. And alternatively, it can also be found in your mitochondria. Good. So now we've got our next question. And here we are working with matching of columns and you will be given a definition, and on the side there will be an option A and an option B. So for this question, yet again, we're going to read, and it's a process of elimination. Are both of them accurate? Are both of them incorrect? Is only one applicable? What is going on? So they will give you very specific instructions in the exam, and you have to follow it exactly to the T when you are answering it. So let's have a look at how to approach such a question. Now, this one will be found in your section A component of your paper. So 2 possible answers will be given. Oop, I quickly went through that. Way too quickly, sorry, my bad. There we go. So when we are looking at it, when answering the question, read the description and each possible answer separately, make a correct tick next to the one that's applicable and a cross next to one that is not applicable. So here you have the instructions, and they will put the answers that you need to focus on here in bold, how you should be answering the question. So typically in the exam, the question would read, indicate whether either of the descriptions in column 1, if it applies to A only, B only, both A and B, or none. So when you write down the answer, they even take it a step further and they say, write A only, B only, both A and B, or none. They provide those specific instructions, so you have to follow it. So you can't say, for instance, A-B or A, B. You have to write it as they've prescribed it to you. So let's have a look at how to answer such a question. Now, here we've got an example of a sex-linked genetic disorder. And when you are at school, your teachers would have told you that sex-linked, those are diseases that are linked to your X chromosome. So when we are looking at males and females, specific diseases that will affect that X chromosome. And you would have learned about hemophilia, and you would have learned about color blindness. So these are prescribed topics that would have been covered. So then you would have ticked next to both of them. They are both correct, but we look at how we should be answering it. So both A and B. So when we write it out, we make sure to follow their specific instructions. Right. Next up, we have our graphs. And here, it's usually a good idea to do a practice run on how to draw your graphs, as this can be quite a tedious thing to do in the exams. If you don't get enough practice, it might take you very long in order to answer this question. But it's important, yet again, not to leave it blank. If you at least have a nice heading and you've got your X and your Y axes, then and labels, of course, then you will at least gain some marks. So the starting point will be to go through 4 different types of graphs, and we're going to look at examples of them shortly. But you would need to know a line graph, a histogram, a bar graph, and a pie chart. So those are the 4 ones that you would need to practice at home. So let's look at the differences between them quickly. So we're starting off with a line graph here, and as we can see, we've got our dots, which you would need to plot, and you basically just connect them with a line. So join the dots, and there we have our line graph. For bar graph, the word bar space graph, so that little space that we see in between the 2 parts to it, this should be an indication to you as to how the graph should look. So I like to use like comparisons and like pictures and all sorts of things when I study. And for this particular example, I remember, okay, the word bar graph has a space in it. So therefore, when it comes to my bars, it should also have spaces in between them. And then remember that when you draw your bars, you have to draw your bars equal in width, and the spaces in between also has to be equal in width. The next one that we have is a histogram. And as with the bar graph, which said that it had a space in between it, we noticed that the word histogram has no spaces in between them. So that's an easy one then. It means that all your bars would need to touch each other just like the word. And here we can see all the bars are right next to each other. So there are no spaces. Your bars are equal in width, and obviously then you will get all your marks for it. And then the last one that we have is a pie chart. And for this type of question, when it's asked in the exams, please make sure that you have your protractor and your compass present. Otherwise, you won't be able to draw a pie chart. So you won't be allowed to swap equipment with one of your friends sitting close to you. And therefore, it's vital that you come prepared with everything that you need: protractor, compass, and a calculator. Right. So let's look at an application quickly of a bar graph, a line graph. And in this example that we have here, they're asking for the relationship between the production of a growth hormone and age. So this is like human growth hormone, and how the production decreases over time. Oops. So we see that trend is a decline in amount of growth hormone that is being released inside our body. So we already know that our dependent variable goes on the y-axis, independent variable goes on the x-axis, and also remember that we have to give our age in years. The unit of measurement that we are applying here is years. So your answer as a label cannot just be years on its own, because we need to give a unit. Right, so how do we draw this graph? So we're obviously going to have to decide on a scale. And in this example that we have, we can see it going up in 200s. Here on the x-axis, we can see it going up in tens. And then, as we said, we can also read off the graph that there's a trend, a decline over time. So when it comes to exam, it will be your job to look at the data set that has been given. So we can see here that they've got plotting points ranging from 2,000 all the way down to like 10 or 20 or whatever that lowest number would be. And you would need to decide, how am I going to make the data set fit onto my page, onto my graph? Because you won't be allowed to just plot the data set as is given in the table. You would need to set out your own scale. So in order to make it all appear nice and not squished together, this person decided they were going to go up in 200. So every 200 or every 5 centimeter, the unit goes up in 200 units. And the same applied for the x-axis. So the person decided maybe every 5 centimeters, the unit is going to go up in 10. And then yet again, the next 5 centimeters goes up in 10. So that will allow for our graph to be nicely spaced out. Right, you could be asked to read off a graph as well. And for this one, they want to know how much of the growth hormone is secreted at 30 years. So we have to go and find 30 on our graph, which is indicated there by the red line. There we go. So there's our 30. And we need to then read it off of our y-axis. So we just draw a line to the side, and we will see that the answer is 800. And obviously remember that when an answer like this is given, that you have to include the unit of measurement. Because the question would be, what if you just had to write 800? Is it 800 apples? Is it 800 pears? What is that 800 that you have just written down? If the unit has been given in the question itself, then it's fine. Then you don't have to repeat it. I tend to repeat it just to make sure that I get that mark. So just be careful for a question like that. Right. When it comes to our calculations, there are some simple math that you might be asked to apply. And as we've mentioned already, make sure that you have your calculator with you. Show all your working out. And this is vital. Everything that you calculate in your paper, put it out step by step and show your final answer, because those steps will also give you a mark in the exams. And then, as we've already mentioned, include the unit. Right, so in this example, and this is a typical example that can be given, is to calculate averages. And I'm sure that when it came to your exams that you might have looked at your reports and had to calculate your own average for all your subjects for that particular term that you were in. So let's have a look at how to do it. If you don't know how to do it already, it's pretty straightforward. All you need to do is take all the marks that you have obtained in all your subjects and divide it by the amount of subjects that you have. So let's apply it quickly. So in this example that we have, we can see that the person's gone and added up all those numbers that we see here. The question wanted to know what is the average pulse rate. So you would have to add up all those numbers for each of the participants. And because there are 5 participants, we divide it by 5. So then we reduce our answer. So that's 440 over 5. And then we get our final answer, which is 88 beats per minute. So remember that we have to then include that unit of measurement. Right, so that's for calculating averages. And the mark allocation that will go with it is for writing out how to calculate it. A mark could be given for that initial step. It will most likely be given there. And then for reducing it, and then your final answer. So when you approach this in a question paper, look at the mark allocation. If the question was out of 4 marks and you only wrote one thing on that piece of paper, then that should be a clear indication that maybe you should write something else on it. So let's have a look at another math. Right, so here we are asked to calculate percentages. And for the percentage, they want to know a plant species found at an altitude of 1,500 meters. So if we go and we find that 1,500 meters, so here it's given, and the number of plants that are present at that point, that would be 60. Now, because we are interested in all of the plants that have been given, we are going to put that 60 over 180 to determine what the question has asked us is to figure out what percentage that particular height the plant present would represent. So we take 60 over 180 times it by 100 to get percentage, and that leaves us with 33.3%. And remember that unit here again. Mark allocation could be for writing out how to do it, the math behind it, and then for your final answer. Right, so that brings us to some simple mathematical questions that could be asked. Obviously, there could be percentage increase questions as well, which you would need to practice. But let's dig into our first example or our first part of the questions that we will be looking at. So we're going to do a little bit of revision first, and then at a later stage, we're going to apply what we've learned in the next slide to some past exam paper questions. So we're going to start with protein synthesis first. And we start off with this image that has been taken off of Mind the Gap. So you guys would have access to it. It's available on the internet as well. And for protein synthesis, like the word says, we are synthesizing. We are making a protein. So the question is, how do we make proteins in our body? And it's pretty straightforward in that DNA will eventually make RNA, or will result in RNA being formed. And from that RNA, we will have a protein that will be synthesized off of it. So that's the short, like, descriptive pathway that it will take. But let's quickly look at what is happening in this image. So here we've got a DNA double helix. As we know, that is the instructions to how our body works, how to make hormones, how to make enzymes, both of those things being proteins. But the DNA cannot leave the nucleus. So in order to get the instructions on how to make a protein, we need to allow the DNA to open up slightly. So weak hydrogen bonds will break, and that will expose the nitrogenous bases that are on the inside here. So there will be a nitrogenous basis of the DNA here at the bottom and at the top. And one of those halves, or DNA halves, will act as the template. So off of this, your mRNA strand will synthesize onto the DNA strand, making a complementary mRNA. Now, this mRNA then has the ability to leave the nucleus because now mRNA stands for messenger RNA. So he's now able to take this coded message out of the nucleus to the site of protein synthesis, which is indicated here. This is the ribosome. So the ribosome will then attach to the mRNA strand, and it will read it 3 nitrogenous bases at a time. And how it's done, how this eventually translates into a protein, is that you would have a tRNA molecule that is known as your transfer RNA that goes and collects amino acids and brings it to your mRNA on the ribosome, and where there's a complementary anticodon-codon match, it will latch on with its amino acid, and then it will start to add to a string. So as soon as that amino acid is attached by a peptide bond to the next amino acid, this tRNA will release, and a new tRNA with amino acid will come into the next 3 nitrogenous bases. So your mRNA is read 3 nitrogenous bases at a time. So pretty much you have like computer code, and computer code is read in 1s and 0s. In our human body, our code is read in 4 different letters, A, C's, T's, and G's. And when it comes to the RNA, instead of T, we've got a U being kicked into the mix there. So I like to think about the process of protein synthesis in various different ways. Now, you would have seen that there is a phase called transcription, and there's a phase called translation. So the word transcribe, scribe means to write. So as soon as you look at your DNA, I think about the DNA as being a recipe book, and that recipe book is in a foreign language. So it's in the library, but you really want to bake that cake because you can see it looks good. So what you will do is you will copy that recipe down onto a piece of paper. So that paper will represent the message, right? The messenger RNA. So now you take that message out of the library and you take it to someone that can actually translate that message. And in our scenario, the translator will be at the ribosome. So that person will take that coded message and translate it over, and it will allow you to actually go and bake a cake. So I say that that cake is now being baked at the translator's house. So this is just a different way to think about it, and to make it a little bit easier. So when it comes to the doctor, even if you go to the doctor will give you a prescription. He would have written that down. They would have transcribed it onto a piece of paper, and then he will go to the pharmacy where you will collect your medication. And that will be where the translation of the doctor's terrible handwriting will take place. So that's just different ways to look at it, to simplify it for yourselves. Right, now when it comes to your examination guideline, your examination guideline would have told you these steps, exactly how to set it out, to write it down in the exam. So transcription, if you learn this like a parrot, you will do pretty well. If this had to be a potential essay exam question. So we start off with our DNA double helix unwinding. How does it unwind? Through weak hydrogen bonds breaking. So between those 2 halves, you will have hydrogen bonds in the middle, and that will form our 2 separate strands. One strand is going to act like a template as the template, so that will be our bottom, actually at the top one in this example, because those little nitrogen spaces are pointing upwards, and an mRNA strand will form. So then, how's that mRNA strand form? Using free RNA nucleotides from the nucleoplasm. So that nucleoplasm is the liquid component of the nucleus, and your RNA nucleotides, these are the building blocks of DNA, and in this example we're making an RNA, so it's RNA nucleotides. Then the mRNA is complementary to the DNA, meaning this fits onto that template strand. It is a copy of it. It's complementary, so if there's a C here, then there has to be a G on our mRNA strand. And then the mRNA has now been formed a coded message for protein synthesis to occur. So that is now transcription. When we look at translation, it goes out of the nucleus to the ribosome, attaches to that ribosome here. We can see our tRNA molecules. There's tRNA, it looks like a little fork. The fork points here at the top, that will be where the nitrogenous bases are located. And this is an anticodon. So, sorry, my bad. This is the anticodon. This is the tRNA molecule. So just to put that into perspective quickly. So each tRNA carries a specific amino acid. So they've represented the circles as the amino acid, goes and picks it up and takes it to your ribosome. It says when the anticodon on the tRNA matches the codon on the mRNA, it brings the required amino acid to the ribosome. And as soon as the amino acids are next to each other, peptide bonds form in between them. So it's important that a singular bond is known as a peptide bond, and eventually we have the required protein which is formed. Okay. The last one that we're going to have a look at is DNA replication. And it's vital to remember that when we talk about DNA replication, it is completely separate to the process of protein synthesis. There might be overlapping words that we will be using to describe the process, but they have nothing to do with each other. Protein synthesis makes protein. DNA replication, like the word says, we are replicating, we are forming more DNA. So the question is, under what circumstances would we want to form more DNA? And pretty simple, when you scratch yourself and you've got a scab and you want the skin to heal over it again, then that will be where new cells would need to form. So during the process of, for instance, mitosis, for the healing process, you would need to form new cells which are identical to the pre-existing cell. So therefore the DNA will be replicated. Now let's have a look at how this process works. So yet again, we're going to find that our DNA double helix, it's present here, but this is going to completely zip open all the way to the top. And what we will find is that both of these strands will now act as template strands, where originally in the previous story they spoke about one strand only. DNA replication, both strands will be replicated. So let's have a look. It says the DNA double helix unwinds and the double-stranded DNA unzips. How does it do it? Through the weak hydrogen bonds breaking, and then as each original DNA will then be serving as our template. Free, yet again here, DNA, because we are building DNA, nucleotides, build a new DNA strand onto each of the original DNA strands by attaching their complementary nitrogenous bases to each other. And here it's important to go and write what are those complementary nitrogenous bases, and that will be an A binds to a T and a C binds to a G. This results in two identical DNA molecules. So I have a silly way to remember the complementary nitrogenous bases. If you make a C, then obviously we all know what a C looks like. But when you make a G, you kind of start off as a C, and then, you know, you've got that last bit, which is the G part to it. So you need to make half a C in order to make the letter G. So that's how I remember. C goes with G, and A goes with T then, which is the opposite. So that brings us to our last little revision slot for this section. And soon we will be looking at a few exam questions. So see you guys later.